In this short presentation, I'll provide an overview of the biomembrane sensor and an accelerated release environment developed with the Safety by Design of Nanomaterials project. The purpose of the biomembrane sensor is to give an understanding of the interaction between toxic substances and a biological membrane. In principle, an electroanalytical technique is performed using a mercury and platinum working electrode hosted by a microfabricated chip. The chip is housed in a flow cell designed to channel the flow of analyte over the working electrodes. Initially, phospholipids are injected into the flow cell, where they form a self-assembled monolayer on the surface of the mercury. The sample is then injected, and its influence on the monolayer is observed by a voltage ramp to measure the capacitance between the mercury electrode and a silver chloride reference electrode. If the sample damages the monolayer, the voltage peaks change shape, indicating that the sample is harmful to biological membranes. The biomembrane flow cell has a layered construction. Silicon and acrylic layers are used to form a sealed flow channel to direct the flow of phospholipid, electrolyte and sample over the sensing chip. An electrical connector connects the working electrode on the sensing chip to a potentiostat. Mercury is deposited on the sensing chip electrodes, which are exposed to the analytes in the flow chamber. An acrylic top plate has threaded ports to provide an interface between the analyte and reference and working electrodes, along with ports for inflow and outflow. Alongside the biomembrane sensor, we're developing a continuous flow release environment to accelerate leaching of nanocoatings, while studying the influence of pH and temperature on release rate. The mini-release accelerator is a flow cell with a layered construction similar to the biomembrane sensor. It houses a material coupon, which is exposed to reagents, and a silver chloride reference electrode, which provides indication of release from the coupon surface. The pH of the supply feed is controlled in a stepwise fashion by a flow selection valve. A flow splitter divides the reagent evenly between corrosion cells to synchronously run sample repeats. A silicon heater pad controls the temperature of each corrosion cell as required. The leachate is collected in a bottle, which can be banked periodically and later transferred to the biomembrane sensor for out-of-line screening. By modulating pH and temperature in this way, a range of corrosive environments can be simulated, including the naturally occurring acidity and basicity fluctuations in the human mouth, caused by the intake of different food and drink throughout the day. This animation demonstrates how the mini-release accelerator can be coupled to the biomembrane sensor for inline screening. The production line, in this case represented by the mini-release accelerator cell, fills a sample loop while at the same time biomembrane sensor is supplied by an inert electrolyte. The sample loop can then be switched into the feed line for the biomembrane sensor and is flushed through to perform the screen. If the sample interacts with the biomembrane, the capacitance peaks on the voltammetry trace diminish. Then, to prepare for further screening, the peaks can be reformed by the injection of phospholipid into the flow cell.